Okay, wonderful. Yes, recording in progress. I have the distinct honor to introduce to you tonight Professor Clarice Aiello. She's a professor at the University of California in Los Angeles now since 2019. He joined that physics department.
Sorry, I need to interrupt. I started in like November 2019, which is not ideal if you're in the business of building experiments.
I see.
Sorry. Sorry. So we're trying to build experiments since November of 2019. So sorry, I close parentheses. Sorry.
Wonderful. No, thank you so much. Thanks for clarifying that. Okay, I'm admitting more people who are joining us. That's great. So yes, Clarice is a quantum engineer interested in how quantum physics informs biology at nanoscale. Clarice is an expert on nanosensors harnessing room temperature quantum effects in noisy environment, which is amazing because we know that quantum effects, we usually need very, a special environment like nanokelvin to see them, but you managed to observe those effects in room temperature. That's great. Clarice, okay, she received her PhD from MIT in electrical engineering and held a postdoctoral appointments in bioengineering at Stanford and chemistry at Berkeley. All the great stuff in all the best schools. So wonderful, Clarice. Thank you so much for joining us tonight and telling us about, quantum biology in nanoscale.
Awesome. And like, since we're a cozy group, can I ask people to like say hi and tell me a little bit about yourselves if you're.
So yeah, people, please feel free to turn on your mic, turn on your video, and introduce
just say
yourself.
Hi. Yes, and like, what's your background and.
Hey, Yusheng.
Hi, Yusheng.
Hi. I'll probably introduce myself first. So I'm a material scientist currently in the University of Maryland. I'm doing computational work on materials and quantum materials, and those materials are part of my project, my interest as well. So that's why I'm here. And I know there are many applications of those materials in sensors. So yeah, that's pretty much.
Thank you, Yusheng.
May I ask which university you work at?
Yeah, it's the University of Maryland.
Fantastic. College Park.
Yeah, that's right.
Amazing. I used to live there for like 6 months. Okay, great.
Wow. We have a little bit snow today as well.
Someone else would like to say hi? Just. Sometimes I think people are afraid of me. I must be very scary. I must have a scary face.
I volunteered Farhad to say hi. How about that, Farhad? Salam.
Hello to you, Reza, and hello, Professor Clarice. Nice to be here. Can you hear me, Reza?
Yes.
Yeah.
Yes, we hear you. Perfect.
I'm very happy to be here, and I'm now to hear from you and to know more about the quantum biology, a major that I fall in love with her.
Nice.
And I love it. And so I want to change my research onto this field of study. No, my research is about the design terahertz sensors, and also I worked on the terahertz biosensing of yes, viral and bacteria.
Cool. So that's very cool because Farhad and Yusheng talked about sensors. So we're going to talk a lot about sensors and like that's fascinating. So I'm going to start sharing my screen And what I'm going to please ask you is to feel absolutely free to pause me so that I can ask questions. I am not sure if I can see the chat box. Where's the chat box gone? Reza, can you, if there are questions in the chat, can you please let me know? I think I see.
Any questions? Yes, absolutely.
In the chat. But
Yeah.
but people, feel free to just unmute yourselves and ask me questions. Let's make it very informal, right? Let's make it a cozy conversation in a cold winter night or a cold.
Wonderful.
It's not that cold here, but it rains, so it's like rain is as bad as it gets in California. So it's also cold here. Okay, so I like to call myself a quantum engineer. This means that I build apparatuses to study and control things that are so small and so well protected from their environment that they're better described by the laws of quantum mechanics as opposed to the laws of classical mechanics. So people get a little bit surprised when I tell them that I study things that might be going on inside birds, inside butterflies. So it's sort of weird, but before I explain where I'm going with my research, I really need to tell you where I come from. So we're going to start talking about hardcore quantum mechanical things. And by the end of the talk, we will have talked about things of biological relevance, such as organismal migration, how our cells respond to oxidative stress, how our cells interact with radiation. And I hope to have convinced you that I think we can learn with nature how to build better technologies. And that's because I'm a quantum engineer who's interested in how quantum physics informs biology at the nanoscale. So I start all my talks by saying that I think humankind is sort of obsessed with measuring things better because it might mean that we understand things or nature better, right? That's this whole deal of making better sensors. So one can think, for instance, of measuring better frequencies. For example, this complicated machine is an atomic clock that sits at the US National Institute of Standards and Technologies, and this atomic clock uses atomic transitions to define very well what a second or a hertz is. We can think about measuring better magnetic fields so that the image of your baby is better resolved. And yes, this is the magnetic resonance image of a baby inside the mother's belly. Or we can think about measuring better accelerations with those tiny little accelerometers so that your gaming experience is enhanced. And actually, those tiny little accelerometers are now ubiquitous in all our handheld devices. But the question that I ask is, what happens if the quantity that you want to measure is very, very small? Or worse, what happens if the object causing the quantity to be measured is very, very small? I'm going to argue that in this case, you need a tiny little sensor that can measure tiny little things, right? A tiny little sensor that gets very close to the sample, to the object of interest. If the sensor is tiny, let's make it very tiny. Let's make it quantum. Actually, there is a whole body of mathematical arguments that can prove, I won't deal with them now, but it can be proven that if you use a quantum mechanical object as a sensor, your measurement is improved. In other words, the sensor quantumness enhances the measurement. In my past, I have worked with a technological quantum sensor, right, that actually was an electronic spin in diamond. And I would like to start my talk by describing this very special technological quantum sensor in diamond. So here's the promise, right? A spin in diamond is a magnetic field sensor. So spin, just a quick recap. Spin is a merely quantum property that doesn't have a classical equivalent, and it's as fundamental as mass and charge. Electrons have spin, atomic nuclei have spin, and spin measures how well a quantum object interacts with a magnetic field. Engineers and physicists and chemists usually depict spin with an arrow, and spin up or spin down just mean two different energy levels associated with this quantum property called spin. So the spin in diamond that I worked with, actually it arose in conjunction with a crystalline defect in the crystalline lattice of diamond. Okay? This crystalline defect in particular, it belongs to a family called color centers. And color centers are the crystalline defects that are responsible for the colors of those very nice diamonds that you see there at the bottom of the slide. Color centers are called so because in the presence of light, right? Light usually of an appropriate wavelength, they absorb this light, get excited, and then de-excite by emitting light, by fluorescing. And the particular color center defect that I worked with, you might have heard of it. It's very hyped nowadays. It's called the nitrogen vacancy center. It arises naturally, but it can also be engineered. And it happens when a vacancy, that is a missing carbon atom, sits nearby to a nitrogen, which is the most commonly occurring substitutional impurity in the diamond lattice. When those two things are close, the nitrogen and the vacancy, like you can imagine it as like a big interaction of electrons, right? The electrons close to the vacancy are unpaired. The nitrogen brings an extra electron, and maybe they're like nearby charges that are caught into this mess. But something extraordinary happens when you start calculating the quantum mechanical energy levels of this mess. And it turns out that the quantum mechanical spin energy levels of this thing really look like the spin energy, the energy levels of a single electronic spin. This means that you can look at this crystalline defect, which is atomically sized, and consider it as one pseudo spin. Okay, you can look at it and think of it as a single spin. And the way that we find those defects in the lab is actually quite simple, and it even works at room temperature. What you do is you take a diamond slab and you put it on top of a moving state. And actually, people now make extraordinarily good diamonds for different purposes, but again, this defect occurs naturally, and the very first sample that we worked with was like a discard from a jeweler. So you put your diamond sample on top of a moving stage and you move it around, while at the same time you are shining a laser at this diamond slab. It doesn't need to be a laser, right? Color centers, they work even with ambient light, as you could see by the colors of those diamonds that you had in my slide. But in the lab, we use a laser because it's more efficient and convenient. And the idea is that as you have the laser, the moment that you hit with the laser one of those defects, it's going to get excited, de-excite, emit light, and this fluorescence we can collect. And this fluorescence, that's what the fluorescence looks like at increasing zooms. So in the bottom right plot there, what you see is like a blob. However, it's a very special blob because this blob is actually the signature, the fluorescence signature of one single spin, right? Compare this with, if you have heard of it, with magnetic resonance. If you want to get the nuclear magnetic resonance or the spin electron spin signal from a sample, usually you need many spins, right? But here you're actually seeing you have a very nice way of getting a signal from one single spin. The defect itself is atomically sized, but this blob is much larger because it's a diffraction limited picture. However, it gets better, as if it couldn't get better, but it can. This effective spin has what people in the field call a quantum state dependent fluorescence intensity. This means that just by looking at how strongly this blob is emitting light, you can actually infer if the state of the spin is up or down. So it actually gives us a very nice way of doing quantum readouts on this quantum object just by monitoring fluorescence intensities. But this is what I promised, that I would make this spin into a very sensitive quantum enhanced magnetometer, and here's how that works. I plot there the two different spin energy levels. So in the y-axis you have energy, and what I call 0 and 1 are just the two different energy levels corresponding to, say, spin up or spin down. The energy difference between those two energy levels is in the microwave. It's about 3 gigahertz. Okay? So it turns out that the state that I mark there as 0, it's insensitive to magnetic fields, so that if the diamond is immersed into a magnetic field, that state 0 won't care. However, the state that I mark +1 there is sensitive to magnetic fields, to external magnetic fields. This means that when it feels an external magnetic field, actually the stop energy level gets displaced, gets shifted a little bit by a tiny quantity called delta. And I think there's there's a question, what makes diverse colors for diverse diamond color centers? The mechanism you described seemed to be unitary. So first of all, the different in emitted light has to do with like the energy difference that you have to emit to go from the excited state to the ground state. I'm not sure if you're using unitary in the quantum sense, like reversible. For reasons that I won't have time to talk about, in the nitrogen vacancy center, the fluorescence is not unitary. It's a dissipative process. We do not do coherent control using this optical transition. I'm not sure if that's the question that Arthur mentioned. Do you want to clarify your question, Arthur? Maybe not. Okay, we can talk more later. And then Face asks, what properties must an object have to qualify as a sensor? So I think I might start to answer your question before. Yeah, so a sensor is something that interacts with their environment in some way, and that gives us some information, right? In particular, this sensor, I'm going to show you how, is going to give us information about external magnetic fields. And I'm going to make a point that is actually very important to your question, Faye. Can you go into detail on how a quantum sensor can detect gravitational waves? I do not know how exactly that is done. And actually, people are trying to use NV centers to detect dark matter. I do not know how things are done, but let me give you, like, in the next like 5 minutes, I'm actually going to be talking about how quantum sensing usually works. I don't know like the details about gravitational waves, dark matter, but I'll tell you like what it means to do quantum sensing, for example. Okay, so back to the energy levels of this spin. One level is insensitive to magnetic fields, the other level, in the presence of a magnetic field, it gets shifted, and there's an interaction called Zeeman splitting, like Zeeman interaction, that shows that the amount of shift, this tiny little delta, is actually proportional to the magnitude of the field. So that, and that's the important part for all quantum sensing. The problem of measuring magnetic fields is actually mapped onto the problem of being able to determine a mismatch, a detuning, some people call, a mismatch from a previously known resonance, from a previously known energy difference. I'm going to pause here because this is actually how most, if not all, quantum sensing works. Okay, the idea is that there are some quantities, in this case magnetic fields, but it can be electric fields, temperatures, and depending on the energy levels or in the quantity, like those quantities can cause a energy shift into a previously known energy difference. So the nitrogen vacancy center is a good sensor in that it can give us information about magnetic fields because the experimenter knows how to measure the tunings delta from resonance. And actually, it's like a signal processing problem, really, right? So other quantities might yield other detunings, other shifts of energy, and it all works because experimenters have known for many decades now how to measure like shifts from energy differences, right? So this is how quantum sensing works in general. But there's a catch, and the catch is that everything that starts quantum, that's classical. And that's the reason why we live in a classical world. That's the reason why quantum computers need usually to be kept at very, very cool properties where there's not a lot of thermal energy that causes quantumness to be lost, right? We can only obtain quantum information out of this spin in diamond before it loses its quantum properties, right? A spin that has lost its quantumness is just a tiny little magnet, okay? A tiny little classical magnet. But before that, if you have taken like an advanced quantum class, before that, there is a phase coherence between, if you are in a superposition of 0 and 1, and while there is this phase coherence, before there is a decoherence of energy levels, you can obtain quantum information out of this sensor. The quantumness time, the coherence time of such a spin sensor in naturally occurring diamonds is about 2 microseconds at room temperature and like in bulk in the solid state, like in the diamond lattice. Right now, it's like a whole field of study. People are actually developing better materials to make like better diamonds that have a longer coherence time, because having long coherence times means that you can get your signal for longer. You can play with this sensor as a magnetometer for longer and longer. There's a question now. Is the microwave here, the controlling microwave, adjusting the population of one? Right. Yes. With microwaves, we can do, if you're familiar with the term coherent control, we can make a coherent transfer between spin populations in the 0 and 1 and vice versa. You could have, for example, Rabi oscillations of the spin going between 0 and 1, right? Yes, the microwave, it's just like the energy difference is in the microwave range. In order to control this spin quantum bit, we are going to apply microwaves, and it's correct that the microwaves do not have to do anything with the external magnetic field. We're just trying to measure an external magnetic field. But I was saying that we want to play with this spin as a sensor for longer and longer, right? Because as it dies as a sensor, it doesn't give us like any useful signal anymore. And actually, quantum physicists have known since the beginnings of magnetic resonance how to control spins, how to decrease the strength of the quantumness quenching noise, right? In order to extend the quantumness lifetime of this of those spins. And actually, all quantum bits made of matter can be controlled with engineered electromagnetic fields, with light, with microwaves, right? And the idea is that, you know, a simple crappy analogy is like, for example, you have a swing, right? You have a swing, and you know that if you give kicks to the swing in a particular way at particular times, you can help keep the swing going. With a matter qubit, or this spin in particular, it's going to be more or less the same thing. So we're going to give photon kicks. It's not really kicks. It doesn't work with like momentum transfer or anything, but it's like you give, you excite it with electromagnetic radiation in particular ways so that it lives longer and longer as a quantum object. So I want to show you one example of such spin control using light, using electromagnetic fields in the microwave range. So before I go to the slide that is going to be the most technical of my talk, I'm going to just make sure that you do not lose track of the big picture, which is we want to have a tiny little sensor that can come close to a tiny little sample. And this tiny little sensor can detect magnetic fields, tiny magnetic fields made by this sample. So whatever I say in the next slide, don't lose track of this big picture. So here's just one example of such a spin control experiment. So what we're doing is just kicking this spin with a microwave field that is known in magnetic resonance circles as rotary echo. Okay, this rotary echo is depicted in jargon in this plot to the right. I don't care if you don't know how to read this. Here's what it means. It means that you are applying, you bring like a source of microwave very close to your spin defect, right? And that you apply a CW microwave wave at with a frequency of the original energy difference between the spin states, right? And it's CW, but at particular intervals, you flip the phase of this wave, CW with phase flipping. For this, actually, electromagnetic excitation has been known and used in the magnetic resonance community for 60 years or more now. And it's very interesting because actually you can change the time at which you flip the phase, and one can prove that depending on which period you choose, you can correct for noise of different types. But what's most interesting is that this sequence, this electromagnetic excitation profile, does not correct for shifts from the resonance, right? You are exciting a resonance like the original, you're sending microwaves at a frequency that corresponds to the original energy difference of the spin, but in the presence of a magnetic field, there's a small shift. This excitation will not correct for those shifts from the resonance. And that's important because if you remember my last slides, shifts from the resonance, the deltas, is exactly what's going to give you information about the magnetic field. So when you do this, you apply this microwave onto the nitrogen vacancy center, this is what you get. You get a signal, which in this case is just normalized fluorescence intensity as a function of the time during which you are applying this tailored microwave field. So here's what this means. It means that when you are at the top of your signal there, you are, say, in one spin state that fluoresces at a certain intensity. If you are at the bottom of the signal there, you are at the other spin state, say spin down, and every signal intensity or light intensity in between means that you are in a superposition of spin up and spin down. For those of you who know what have seen things like this is just like a modulated Rabi experiment. So we can then do a mathematical trick called average Hamiltonian theory, and we can estimate to first order for very tiny energy shifts delta, how the signal depends on this detuning delta that we want to measure, right? And it's a complicated expression. It doesn't really matter, but you see that the signal has a certain dependence, a well-defined dependence on delta. So with this formula, we can actually Fourier transform the signal and get this plot, which is just sort of like a spectrum. It doesn't matter how I call it. And knowing how the signal depends on the delta, we know how to read this spectrum. And I'm not telling you how to read this, but with this spectrum that you see there, we are reading three different deltas, 3 different energy shifts, which correspond to 3 different magnetic fields, the smallest of which corresponds to about 1/10 of the magnetic field of the Earth, which is already quite tiny. But again, not important. Important thing is that you have tiny little sensor that can come in close proximity to a tiny little sample and yield very good quantum enhanced information for some time. So before I get to the questions and make a definite pause, I'm going to make my point here that I've just shown you a quantum sensor that works at room temperature and in noisy environments. It's an absolutely widespread used technological quantum sensor. And again, before I address the questions, I want to leave those of you who do not have a question with a question. I'm going to ask a question. So I've told you that this energy difference between the spin states was about 3 gigahertz, right? But I also told you that this sensor works like at room temperature. This means that this sensor is immersed into a thermal bath, right? For those of you who don't have a question, I would like you to Google how many gigahertz corresponds like thermal energies at room temperature, more or less, order of magnitude. Just I'm asking this in this particular way because I want to make a comparison. But meanwhile, I'm going to read. There is one question. It seems, Farah, do you want to ask your question, or do you want me to read your question? Because I'm not sure I understand your question. It seems that the uncertainty principle restrict a quantumness time. I'm not sure I completely understand, but I don't think it's necessarily so. The idea is more that what causes decoherence, or what causes the loss of quantumness, is more interactions with like the environments, right? So this is sort of why we live in a world that looks classical. Like usually quantum mechanical phenomena are exploited like at very low temperatures in vacuum chambers where there aren't many like molecular collisions. So decoherence usually comes by thermal means, or for example, by interactions with other spins, right? Other spins nearby the sensor spin that causes it to sort of not evolve in the most organized way, if you will. Other questions? In the absence of questions, someone says it's 0.026 eV. Okay, so that might be true. In gigahertz, thermal energies at room temperature are about 6,000 gigahertz. Okay, and I just wanted like in gigahertz units to make the comparison. So this difference of energy, right, between the spin states is 3 gigahertz, and it's immersed into a 6,000 gigahertz thermal bath. So people ask, so what the hell? Isn't this going to thermalize? You know, there's a lot of heat going on around this sensor. How in hell does it work, right? There's this huge energy difference there. And the thing that I can say is like, actually, it is going to thermalize. And thermalizing is sort of very similar to losing the quantum character, right? So after a certain time, that is more or less 2 microseconds, the spin is sort of classical, right? The spin is no longer quantum. And my point here is that even though the thermal bath around the sensor is very, very large, there is still a finite time before thermalization, before the coherence, where the spin can provide us with quantum enhanced information. After thermalization, after the messiness of the environment kicks in, we can no longer obtain quantum information. In the plot of the previous slide, for example, the signal as a function of time would actually just flat line, and then it stops providing us with useful information. So there's nothing mysterious to the fact that the energy levels of the sensor are much smaller than the energy of the thermal bath. And I'm telling this to you using the nitrogen vacancy center as an example, because you might have the same question when I talk about the sensors that I really want to talk about, which is the second part of my question of my talk. So up to maybe 5 years ago, like the NV Center, I mean, those were the type of sensors that I was used to dealing with, right? Like technological quantum sensors. But at some point, it dawned on me that many times, sensors made by nature, be them like either quantum or classical, many times outperformed humankind-made sensors in crazy ways. I'm going to talk to you about a natural magnetic sensor that I'm going to argue is quantum enhanced, and actually that work in a way that it should remind you a lot of the technological quantum sensor that I have just described to you. But before we talk about before we talk about the physics, let's talk about biology at the nanoscale, namely the chemistry. So it has been known, like in the field of, it's called radical chemistry, for many decades now, that magnetic fields can alter the final products of a class of chemical reactions that depend on spin. So to cut a long story short, here's what happens. There's a chemical reaction happening, and at some point the chemical reaction comes to a crossroads, and there's a certain spin of interest. One, like usually it's two spins, but I'm going to make it simpler. I'm going to talk about one spin. Chemical reaction comes to a crossroads, and there's a spin of interest, and depending on the electronic spin state, that is, if the spin is up or the spin is down, the chemical reaction continues through two different branches. If the spin, and here I'm going to use jargon, if the spin is measured by the environment to be up, the chemical reaction continues through one branch. If the spin is measured by the environment to be down, the chemical reaction continues through another branch. Importantly, the macroscopic final products of those two branches are different. So a finicky quantum phenomena, right, of a spin at play, and actually magnetic fields can alter the probability that we find the spin up or down. This finicky, very fast spin flip phenomena can actually have macroscopic consequences and at much longer time scales, sort of like downstream from this injunction at the chemical reaction. This actually has been demonstrated at room temperature, in solution in the gas phase, in the solid phase, and for magnetic fields as small as the magnetic field of the Earth. The magnetic field of the Earth is really tiny. It's about 50 microtesla, which is smaller than the magnetic field that you sense from your mobile when you hold it close to your face. But where does bio enter this, right? So bio enters this field because of birds. We're going to start talking about birds. Birds are interesting, but I'm going to be so happy because there's going to be a slide where I'm going to stop talking about birds. But we need to start talking about birds because historically that's how it happened. So again, it's been known, like biologists have known for decades now, that birds, migratory birds, when they migrate, it's sort of known that there is no doubt that, at least as a partial cue, birds do use the magnetic field of the Earth to migrate, right? And people have always tried to explain how. People have made models like, oh, do they have like a tiny magnet in their beaks or something? There were a lot of theories, but no theory could predict the behavior that people saw, like on how birds migrate depending on magnetic fields, right? So at some point in the end of the 70s, some brave theoretical biophysicists made this outrageous hypothesis at the time, right? They were people thought they were crazy, right? They were like, Well, listen to me. Were the same type of spin-sensitive chemical reaction, were those reactions to be happening at room temperature in the messy conditions of like an organism, a living organism, birds and organisms in general could sense magnetic fields to the extent that they could sense different physiological products coming from those spin-dependent chemical reactions. So, for example, they posited that maybe, maybe there's something in the eye of the bird that that modulates the photosensitivity of the birds as they look like left and right, so that if magnetic fields have an effect on like the photo modulation of the sensitivity of like how much light they're seeing, maybe birds looking left and right could actually see magnetic field lines and know where they should go. Right? Whereas this particular hypothesis has not been proven or refuted yet, nobody really knows what's going on in birds. People really started looking for whether, if those chemical reactions were happening, where they could happen chemically. And people started looking at animal proteins because birds, right? But at that point, the only animal protein that was known to be able to sustain spin-dependent chemical reactions was this flavoprotein called cryptochrome. So cryptochrome is a flavoprotein, has a pigment flavin. It's a fluorescent protein, that is, it absorbs, in this case, blue light. It gets excited and then emits light back. Birds have cryptochromes in their eyes. Migrating butterflies have cryptochromes in their antennas. And actually, we have cryptochromes also in our eyes. And biologists tell me that this statement is absolutely good. We have cryptochrome in all our cells because cryptochrome is also involved in circadian rhythm regulation, right? And actually, this is not crazy science, but if you change time zones, if you take an airplane and fly and like suffers jet lag, people say, well, you can get better from the jet lag if you look at blue light. There are those like Philips lights, like blue lights that people say can help you with the jet lag. Why that works is because you actually put your, I don't know exactly how it works, but it sort of kicks up your cryptochrome. It puts your cryptochromes to work somehow. And it's not me who tells this. This was told by the guy who won the Nobel Prize, I think in chemistry in 2015, that discovered, among other things, like cryptochromes. Right? And it turns out that cryptochromes are expressed throughout the tree of life. All those organisms there make cryptochrome naturally. And actually all those organisms boxed in red are organisms for which there has been some study, macroscopic study, correlating cryptochrome with sensing the presence of magnetic fields. And given all this evidence, I'm really convinced that there's a lot of things that corroborate the fact that cryptochrome might be mediating the sensing of magnetic fields by organisms. But unfortunately, the existing evidence is very disconnected in length scale. So there's evidence at like the macroscopic length scale to the right of my slide, and there's evidence to the left of the length scale, like in basic chemistry. So in basic chemistry, so in the evidence to the left, there is there is sufficiently good evidence that shows that cryptochromes are actually working as very good quantum sensors of magnetic fields. For example, in the top left plot there, what you see is the result of an experiment where people took a vial, a test tube of cryptochromes, and tracked the fluorescence emitted by this cryptochrome as a function of the time during which they applied a laser, like blue light, to excite this cryptochrome, right? Cryptochrome absorbs blue light, gets excited, and fluoresces. The first thing that you see is that this fluorescence is going down. You're photo bleaching your pigment. It only means that the laser is sort of killing your sample. But as the researchers post a tiny magnetic field on and off, as they were doing this experiment, what they saw was a tiny modulation of the emitted fluorescence that you can see in the inset there that followed the magnetic field profile that the sample was subject to. This is not an artifact. This has been reproduced with different magnetic field profiles. And, this is in very close analogy to what we had seen for the nitrogen vacancy center. Remember, just by looking at how strongly that nitrogen vacancy center that blob was emitting light, we could infer if the spin was up or down. Here, it's sort of the same thing. Just by looking at how strongly the cryptochrome sample is emitting light, you can actually infer whether the spin was measured by the environment to be down and the reaction continued to the down pathway, or if the electron was measured by the environment to be up and the chemical reaction continued through the other pathway. Moreover, there is not definitive, but good enough evidence that shows that the electron of interest in cryptochrome can be quantum for up to at room temperature in solution for up to about 1 microsecond, which is again on the same order of magnitude that I quoted for the naturally occurring diamond, right? So all of this is very good, except that the next step on the experiments, when we have birds, we go from like proteins to birds, right? And this is very complicated. Those experiments are very hard to do. People have like 1 month to do those experiments. Like during migration season, they work with like 20 birds each year. They put the birds in cages and then they monitor which way the bird wants to go out. And if the bird doesn't see light that corresponds to the absorption band of cryptochrome, the birds do not orient, right? They cannot see the magnetic field. If people put coils around and change the direction of the magnetic field, the birds change the way that they want to go out. It's all consistent with the spin phenomena. For other thing, flies can apparently, this is like magic to me, flies can be trained to find food via the presence of magnetic fields. So researchers can do this somehow. And then the researchers knocked out the cryptochrome gene of the flies, and then the flies were no longer able to find food via the presence of a magnetic field. In a further experiment, the researchers put back in those mutated flies human cryptochromes, and the flies were back to being able to find food via the magnetic field. Right? Again, all of this shows that there is a dependence on cryptochrome, but it's very hard to connect chemistry to like behavior of organisms. So here's where we enter, right? We want to start bridging those length scales. So what we want to do is quantum-inspired experiments in proteins inside, say, one cell, a couple of cells. We want to take a bottom-up approach, right? And see if we can, for the first time, recover or not the fact that, say, cryptochromes are indeed working as quantum sensors inside a cell, right? Read out, like see if we can change the spin state of the cryptochrome, right? And we can see the state of the cryptochrome by fluorescence, and then compare this state of the spin to what happens downstream in a cell. In other words, what we build are glorified microscopes with coils. The coils to mess up, change, and control the state of the spins, and the microscope to read out via fluorescence the spin states, right? Which happens very fast from nanosecond to microsecond timescales, and compare that to usual microscopy timescales, like in cells with usual markers, which is like millisecond sort of timescales fluorescence. So when I talk about microscopes with coils, the instrumentation is not going to look like the the microscope to the left. It's going to look like the setup to the right. This is not our setup. This is a picture, a random picture that I got from the internet of like an atomic physics experiment. We're going to have an atomic physics, a quantum sensing experiment that works on a biological sensor, on a biological sample, right? But really, I think that this research inserts itself in the field of called quantum biology. And I really think that the current bottleneck in quantum biology is the lack of quantum inspired high-tech instruments where we can probe quantum states in biology, right? So this is our approach. We want to push the envelope and start using quantum technology we want to treat those spins in biological sensors again as bona fide quantum sensors. We want to apply electromagnetic fields to control them, use light readout to read them out, and do quantum inspired experiments onto biological samples. And what's different in our approach from usual biophysics experiments is that in some biophysics experiments, you observe something and then you model. Here we have the dramatic advantage that we can actually use quantum mechanics to model those things. The minimal quantum model to model what's happening in Cryptochromes, for example, involves simulating tree spins, that is, solving the Schrödinger equation in the presence of noise for a system composed of three spins. I can give details later, but it can get way more complicated. My theorist colleagues can mimic like 21 spins and things like this, but we can actually use quantum mechanical tools in conjunction with parameters that come from the photophysics, the chemical photophysics of, say, Cryptochrome, photophysics, its redox cycle, and we can start taking those things, plug in numbers, and make predictions. I'm going to show you one prediction. It's like, forget, there's 4 plots there. I only want you to look at the top right plot. We can start making predictions that are yet to be taken experimentally, right? The problem of current experiments in the area is that there's not a lot of systematization of like people rastering magnetic fields. So here's one prediction that I would love to take experimentally. What I plot into the top right plot is the magnetosensitivity of this cryptochrome protein, right? How much it's sensitive to magnetic fields as a function of the magnetic field strength that it's immersed in. The first thing that you see is that this magnetosensitivity is not monotonic. It goes up and down. At like 10 times the magnetic field of the Earth, at about 600 microtesla, it has already decreased by a lot, which is extremely important, which means that strong magnetic fields do not enhance the effect. It means that putting these molecules of interest in a nuclear magnetic resonance machine, like a couple of Tesla, is not going to happen. This type of spin physics is eminently a weak magnetic field phenomena, which is also a problem because all those things emit relatively weak magnetic fields. The second thing I would like you to show, to see in this magnetosensitivity curve is that it peaks, the way that the place where it peaks is actually very close to the magnetic field of the Earth, which is either it's either a coincidence or it shows that there might have been some evolutionary pressure for those molecules to work best under the magnetic field where they developed, right? We cannot tell those things apart, but we think that people who do directed evolution of proteins could help us see whether we can put artificial evolutionary pressures, like magnetic field pressures onto proteins of this type. But I'm ready to stop talking about birds, and this is almost the end of my talk because my research would be much, much poorer if birds are okay, but they're not everything in life, right? So birds are really okay, but the implications of this research seem to be much broader than what was previously thought. There have been many recent results, again, most at the macroscopic level, say with plates of cells, showing that metabolic processes such as production of reactive oxygen species, respiration rates in cells, glycolysis rates, acidification rates, cell regeneration growth, magnetic DNA repair, all those things seem to depend on weak magnetic fields in a way that is consistent with a spin model. I want to spend some time talking about those two papers there. The first shows that cellular autofluorescence is sensitive to magnetic fields. Those people are probably seeing the fluorescence of flavoproteins in mitochondria. So mitochondria are like extremely important, right? So if there's any important spin phenomena there, we want to know, and we want to control it. And the most extraordinary thing that I hope you're going to get a kick out of is this lower picture there. So here's the story behind the picture. I have a friend who's like a hardcore precision measurement physicist, and for a living, he builds hypomagnetic chambers. So those are chambers that protect very well against like external magnetic fields, and it shields even the magnetic field of the Earth. So the magnetic field of the Earth is 50 microtesla. Inside his chambers, there's like a DC noise of 1 nanotesla only. And what he usually do is like put like cold atom experiments inside, and he does like precision measurements. It doesn't matter. But he started hearing about those magnetic field effects. So here's what he did. And now this experiment has been, it's not published yet, but it has been reproduced by another group in Mainz and another group at Tufts University. He grew tadpoles inside those hypomagnetic chambers under 2 conditions. The first, they put tadpoles, like frog embryos, inside for a couple of days, because that's as long as they could go without asking for a permit or something. In one condition, they grew the tadpoles with a mimicked magnetic field of the Earth applied inside. And in the other condition, they grew the tadpoles with just the nanotesla magnetic field residue. For the tadpoles grown under the mimicked magnetic field, they were macroscopically sort of okay. But for the tadpoles grown under like the nanotesla residual field, 30%, as you can see in the picture, were macroscopically deformed. Unclear if they're viable, they're absolutely deformed. And whereas that does not prove at all that this is spin related, given the magnitude of the magnetic fields involved and the fact that you're not even giving the magnetic field, you're only taking out the magnetic field, everyone's best guess is that this is spin dependent. Right? Again, it's not only cryptochrome, melanin, which apparently is what gives the tadpoles their color. Apparently, melanin also has very rich spin physics. So there's a lot of things going on, not only in cryptochrome, but having to do with reactive oxygen species, DNA repair, enzymes, melanin, right? Which gives rise to all sorts of crazy questions, right? So like, what's the magnetic field in Mars? Right? Can we grow tomatoes in Mars? Can we reproduce in Mars? So there's all sorts of things that might be a consequence of spin physics in biological systems. So here's what we're setting up to do. We want to either establish or refute down to a certain noise level, right? This also helps the science move forward. We want to either establish or refute whether quantum mechanics can macroscopically alter or can macroscopically be responsible for physiology. And we think that therapeutically, for therapeutic and diagnostics, we think that maybe not in 5 years, but maybe in 25, 50 years, we make this from a science problem into an engineering problem, right? Maybe we understand exactly what's going on, so we can start applying weak magnetic fields to tweak physiological parameters, like tweak chemical reactions as we need, to change the course of disease or something, like produce more or less reactive oxygen species, for example. And I really think we can learn with nature to build better technology, because if nature is using quantum mechanics to function, it knows darn well how to deal with noise. And noise is the reason why we have the coherence, right? Why quantum computers do not live forever. So I'm not saying like, let's replace Google's beautiful quantum computer with a molecule, but rather, are there strategies used by nature that we can adapt and apply onto existing, say, quantum computing architectures, right, to make better quantum computers. We're actually not the only people thinking about pushing the envelope on the technology to deal with those endogenous quantum degrees of freedom of nature. All those very recent papers, relatively recent, 17, 19, all those people are taking a very hardcore quantum approach towards biological systems. And I think that those approaches are going to unlock a huge power on how to actually understand and, importantly, control biological process. So I want to end up by saying that I'm crazily thankful for my group. They, as I mentioned, they were brave enough to start a group in the middle of a pandemic in a subject that is many times that goes misunderstood, right? Quantum biology is not mainstream science. And I think that's what I wanted to tell you. So I would like to end up by saying, may the quantum be with you. What are your questions?
Thank you so much, Clarice. Thank you very much for this amazing talk, packed with so much information. I loved it. Thank you. And best of luck to you and your group, and good luck growing group and doing amazing research there at UCLA. We are looking forward to hear, great news about your outcomes and your accomplishments soon. I'm sure we will hear that. Thank you. I think Arthur is asking if we can basically build quantum computers using NV centers at room temperature. Is that a possibility?
Yes, there are many. So there are many people who are actually trying to do this, right? Like there are several flavors of quantum computing. Nobody knows which one is going to win, if at all one is going to win, right? I don't think I have it in my slides, but what people think about doing, and people have started to do this, is like people can implant nitrogen vacancy centers in particular places in the diamond, right? And what people envision doing is use the electrons from the nitrogen vacancy center as information buses, put the defects close enough together so that the electron spins can talk, and use nuclear spins for information storage, right? Nuclear spins are very good as memory storages because they interact less with the environment than electronic spins. So electronic spins for information transfer, and nuclear spins as memory units. So yes, people are thinking about this. If you ask me, I would not cast my vote for an NV Center quantum computer. I think NV Center are amazing sensors, exactly because they talk a lot to the environment. They can obtain information from the environment. And I think they're going to be magnetic resonance approaches based on NV centers. Other types of, so there's a company that makes a fingerprinting of things using NV centers. So NV centers are very good sensors. I would not bet my money on quantum computing with NV centers, but what do I know? Now I care about biology. My opinion might not be the most up-to-date opinion. Other questions?
Thank you. Thank you very much, Clarice. And if I may add, just to everything you mentioned, is a huge problem with building a quantum computing out of NV centers will be, engineering them, because they're happening often naturally anywhere they like to happen, and that makes it a little bit hard. So people, if you have any question, please add them in the chat box, or just feel free to unmute yourself and ask, since we are a small group and we have some time for those questions. If not, I have a question for you, Clarice. One of the applications of these NV centers and color centers in general is thermometry. Have you guys ever looked into any of those applications for bio applications?
No, I have not, but you're right. Actually, it's something that is getting very well developed. So what people do, just for the audience, people are people have a diamond slab. They explode this diamond slab with NV centers, and then they sell tiny nanodiamonds containing statistically like 1 NV center per nanodiamond. People then shoot those inside cells, right? And the principle, like inside living organisms, and then it's sort of the same. There is a certain energy transition that gets shifted according to temperature. So you can measure temperature because you can measure this detuning caused by temperature. So that's something that people have been doing. Apparently, this is a very successful use of NV centers. We are actually not using NV centers at all because our whole goal is to bypass the technological quantum sensor, is to address the spins in biological samples like directly, right? We want to bypass the technological part. It's challenging, but we think that we can, in time, we can get there.
Wonderful, fantastic. Okay, I think Yushin, do you have a question for us?
Yes, thank you.
Yes.
Thank you very much for the nice talk. I actually have a question on the microwave control of it. I know one of the reasons that's quantum computing or the other sensors using NV center, why this is challenging is because it's hard to use the microwave to control, and also hard to place two NV centers exactly where we want them to be. And my question about the microwave is, can you have a like comprehensive description of how hard it is to control? I know one thing is because we're talking about sending microwave to NV centers on the scale of nanometers, so definitely you're going to have some diffraction of the microwave signals you're sending. So you're shining microwave from a field of NV center instead of one NV center. Am I correct on that?
It depends on what your sensor looks like. If you have a slab of diamond with many NV centers, yes, you're going to be exciting sort of everything, right? But remember that you can focus to only get light from a single one, right? If you have only your blob, your fluorescent blob, if that's the only one that you see, you're only collecting light from that. So your signal, even though you are exciting more than more centers in principle, you are only collecting information from one generally. So I think related to your question, and I think things are progressing, but in my time, the simplest way of applying microwaves was just to put like a tiny 25 micron copper wire on top of our diamond sample, and then you just push a current through, and it makes like a magnetic field, right? That's how we excited our NV center and everything around, right? Okay. And microwaves in general, and you didn't ask this, but it's also important, right? It's unclear what it does to the cells. People say that the cells survive, like the microwaves that are applied onto the NV centers that have been shot inside the cells. But yes, the byproduct of such experiments is that the cell is also getting bombarded by microwave, and I'm not sure how much that is and what that does.
Okay, thank you. Yeah, I think that clarifies. So basically, it's you have large microwave field, large EM field you are applying, but you only collect the data information from one specific NV center.
Yes, you can. And I forgot to say that you can also, I didn't mention this, but there are people doing sensing with ensembles of NV center, with a sample that has many NV centers, and there you collect you collect signal from an ensemble of NV centers. But in the cases that I showed here, and usually what's being done in biology, where you shoot nanodiamonds inside a cell, it's usually for one NV center.
Okay, thank you. Wait, I may have another question.
Yes.
Related question to it. So, at the beginning of your slides, you're showing that you're collecting single photon signal. So does that mean you have to decrease your microwave intensity so that you are receiving exactly single photon signals?
So a single NV center is a single photon emitter. And actually you can prove this by doing correlation measurements. This means that you collect your photons, right? And then you push them through a beam splitter. That is, there are two detectors. One, the photons that you get from the NV center can either go through one detector or the other detector. And then you can make statistics of the timing, like that the photons arrived. Usually with such a setup, you can actually prove that you are dealing with a single photon emitter. That is, only one photon is emitted at any single time, right? However, the strength of the microwave doesn't have to do with how many photons you get back. The strength of the microwave, the power knob in your microwave source, has to do with how fast you can control your microwave. It has to do, if you've heard of this term, it has to do with the Rabi frequency of transfer. It has to do with how fast you can transfer the population coherently between the up and the down states. Actually, if you can put more microwave power, it's better, because this means that you can actually do more things with your sensor before it decoheres. Some people, like a very crude measurement of like how, like if you have a quantum computer that decoheres after time X, what you want to do is like how many quantum gates you can perform in this time X. Right. So with the NV center, it's sort of the same thing. If you have a faster Rabi frequency, you can have like you can control and do things with your sensor faster, so that in the end, it's not that you get more signal, but you get more. You get. It's not more information, but you can induce more transition. I'm confusing myself here. All I want to say is that many times it's good to have very large Rabi frequencies. It's everyone is aiming to have as high power of microwave seen by the sensor as possible, as generally. Right, that's what everybody wants, because this means that you can have faster control, in the sense that it takes less time for you to do a spin flip, for instance.
Great, thank you very much, Clarice. And yeah, basically NV centers or color centers are intrinsically single photon emitters, right? In each transition, they're only sending one signal, so it doesn't matter what is your, the intensity, let's say, of the microwave. Thank you so much, Clarice. I see, Arthur, you are on video now. Do you have a question? All good. Thank you very much. Okay. We are like 15 minutes.
I have a question, Reza.
Sure. Go ahead, Farhad.
Thank you for your enthusiastic presentation and discussion, Professor Clarice. I have a question about the quantum time.
Yes.
I want to know that how long quantum time could increase. Okay, and because I am a PhD in I have PhD in molecular and atomic and molecular physics.
Okay.
I have a question about that related to quantum time, about the uncertainty principle. Do you know that you have two quantum state for a spin down and a spin up, okay? And you applied the static magnetic field on, for example, on diamond vacancy, okay? And because of the Zeeman effect, the upper state shifted about delta in energy window, okay? And according to the uncertainty principle, I think this excited state have a limited lifetime, okay? And
it's not an excited state. It's just a shift in energy, right? You can imagine this like the zero state. You can imagine it like in an oscilloscope. It's like a 0 volt signal, right? And usually say that the other state has a +1 volt. It just means that in the presence of a magnetic field, this 1 volt signal is going to be promoted to say 1.3, right? And this 0.3 energy difference can give you information about the magnetic field. So it's not an excited state. It stays there. The +1 signal is stable there, right? The +1 energy level it changes to that value.
Thank you, got it. And you think that you said that the quantum time for diamond vacancy was about 2 microseconds, and for cryptochrome protein about 1 microsecond, okay?
Yes.
And what do you think about the increase this quantum time? What we should do for reaching?
So to increase those coherence times, what people do is apply those tailored electromagnetic fields. Right. For example, in that signal that I showed to you as a function of the time during which we applied this continuous wave microwave with phase flip, I don't have the slides, but this the signal it was sort of going down. It was modulated, but it was going down. But I could acquire signal up to like 5 microseconds, right? Which like at 5 microseconds it hadn't decayed completely. So for example, with this we are already extending a little bit those quantumness times. So coherence times can be made longer, or in other words, we can quench the effect of noise by applying those engineered light pulses or microwave pulses, engineered electromagnetic pulses. And that's like that's how the whole field of magnetic resonance works. People play with the spins and make them quantum for longer by applying like pulses of microwave and things like this.
Okay, thank you for your answer.
Okay, wonderful. Thanks, Clarice. And thank you everyone who could join us. This was a really, really amazing talk. I enjoyed it a lot, Clarice. All the best to you. Of course. And yeah, we look forward to having you, later with amazing results and publications from your group and hearing great news.
Awesome.
Thank you.
Thank you. And I'm looking forward to hearing back from you. If you have a question, just feel free to contact me. And thank you for having me, Reza. And we talk soon again, I hope.
Very much my pleasure. Thank you. Yes, if you have any question, Clarice email, and you can find him on UCLA webpage.
Thanks. My email is there's CLA@UCLA. That's also easy. The first 3 letters of my name at UCLA. I'm going to write it here.
CLA. Okay, wonderful. Perfect.
Thank you, everyone.
Thank you so much. I was going to do that. Thanks. Thanks, Clarice. And thanks, everyone, for joining us. Please stay tuned. Follow our webpage. We will have more events like this. And yeah, stay safe and have a great night. Thank you.
Very excellent. Thank you.
Thank you all. Bye-bye.
Thanks, Arthur. Bye-bye.
Well.
Thank you,
Bye.
Clarice. Thank you.
Bye.
Thank you, Reza.
Thanks, Arthur. Goodbye. Bye.